Hello everyone, today is the day 4 of my 7-day read an article challenge. In this case, I am starting to read articles that are scientific articles, and today is the day 4 of this 7-day challenges. By the of the 7 days, I'm hoping that it is going to increase the amount of information I have on the topic of my interest that I have chosen, and it's going to help me with processing of informations, improve my memories, and also make reading of scientific articles fun for me. So this is the 4th day of my 7-day workout. I hope I can continue it for the whole 7 days. Let's see. Now, beginning with today's topic that I'm going to present, it's about the protein channels called chloride ion channels. And the family of proteins are known as CLC. In this paper that I'm going to present today, there are two main CLCs that have been discussed, CLCE and CLCF, about which I'm going to talk. The paper and the affiliation of the authors are listed below, and the link is given in the description box if you would like to read the paper. Now, beginning with the introduction, generally in plants, for photosynthesis to occur properly, there is often importance of channels that can transport ions, and such channels that transport anionic moieties such as chloride or nitric oxide or things like that are known as anion channels. One such anionic channel is the chloride channel family proteins called CLC, and they were first discovered in tobacco plant. In Arabidopsis, there are 7 CLC genes present, and they are numbered from A to G. So ATCLCA means AT stands from Arabidopsis thaliana, CLC stands for chloride channel, and then A is the numbering, and then like that we have from A to G, the final being ATCLCG. The intracellular localization of the proteins can be understood by studying them, by co-expressing them with other kind of proteins such as green fluorescent proteins, and that has been utilized in this paper in order to study the localization of the protein and the functionality of the protein. Physiological characterization of Arabidopsis mutants have suggested that the involvement of ATCLCA and ATCLC in the regulation of nitrate levels in plants, but the role of ATCLCE and ATCLCF are not well understood, and that is why we are reading this paper, because it's going to give us an input on where ATCLC and CLCF are present in the cell and what is the function of these proteins. So if we look at the gene sequence of this ATCLC, ATCLCF, and the protein sequence of ATCLC and ATCLCF, we come to know that this ATCLC and ATCLCF are present in two different chromosomes. ATCLCE is present in chromosome 4 and ATCLCF is present in chromosome 1. This ATCLC and ATCLCF proteins belong to higher plants, that is, they are in the eukaryotic group. And if you look at the dendrogram present on the right hand side of the slide, these CLCs families are pretty homologous to their CLC homologs that are present in prokaryotic organisms such as bacteria or fungi. And that is why it is important to look at the features of these higher plant CLCs. So if we look at the ATCLCE and ATCLCF, they still form a distinct subfamily among the group of the CLCs present. And these proteins have a 12 membrane spanning domains with 18 alpha helices. ATCLCE gives rise to a protein of 709 amino acids with a molecular weight of approximately 75 kilodalton, and ATCLCF encodes two different proteins. The shorter one is a 586 amino acid of approximately 62 kilodalton, and a longer polypeptide of 781 amino acid of approximately 83 kilodalton. If we analyze the protein sequences between the various 7 classes of ATCLC starting from A to G, we observe that most of them have two glutamate residues, E148 and E203. The E203 actually distinguish if the channel proteins are only channels or if they are antiporters. In case of ATCLC A, B, C, D and G, both the glutamate residues are present, whereas for ATCLC-E and ATCLC-F, there is only E148 present, suggesting that the mechanism for transport of fluoride ion through this one is different from the ones reported for A, B, C, D and G. Now we want to look at the localization of ATCLCE protein. So ATCLC's localization was initially tried to be found by studying the Aramemnon database, but where it is exactly located, whether it's in mitochondria or in chloroplast, that could not be properly identified just by the database studying. So then there was a GFP expressed version of ATCLCE that was put into the onion epidermal cells for experimentation, and that gave insights on where ATCLCE was present. So on expressing the ATCLC with GFP in the epidermal cells, the cells glowed up in the cytosolic region and also within the nucleus. If we look at the amino acid sequence for ATCLCE, the N-terminal region that is important for targeting of this protein into different organelles or places has alanine residue present in the second position, and there are too many serines present and proline present, and a total like a huge global positive charge is present on this sequence, which actually favors the localization of this protein into plastids such as chloroplast. The cleavage site that is present in between these amino acids are between the S17 and R18 residues in the N-terminal region. And as shown in the first figure of the expression, you can clearly see the fluorescence in the cytosol and within the nucleus, which is like in bright green in color. And there was also a dotted fluorescence pattern that was seen, and it was restricted to mobile organelles located in the cytosol with a diameter of about 3 micrometer. This diameter is small enough, suggesting that there could be a plastid localization possible. On co-expression of ATCL-CEGFP with another targeting protein that is specific for mitochondria and plastids called SYTP-DS-RED, there was a revelation that when both of them are present in the same organelle, in that case it would give a yellow signal, whereas only if one of the signals were present, you could see only the green color or the red color, as you can see in the panels C, D and E. On transient expression of ATCL-C GFP construct in the protoplast from Arabidopsis cell suspension, the colocalization of the fusion protein of the GFP giving green fluorescence and the chlorophyll fluorescence showed in the red fluorescence was observed, demonstrating that the chloroplast targeting of ATCL-CE. To further analyze that if it is definitely in the chloroplast or not, as seen in from the panels F to H, there was an immunoblast that was done, or also we can call it as a Western blot because we were trying to see the protein over here. There was an antibody raised that was against an ATCL-CE, or it was against anti-CLCE along with IgG. Using both of these antibodies, the CLCE was targeted in order to see where it is, which fraction of the chloroplast is this protein going to go and be present. So on the experiment done with various fractions from the chloroplast, it was revealed that this CLCE protein is present in the chloroplast, but also it is present in the thylakoid and not in stroma or the envelope fraction of the purified chloroplast. Based on the transcriptomics data that was generated from this software called Gene Investigator tool, it was seen that ATCLC expression is higher in green tissue, and also it is higher in cotyledons in seedlings rather than in roots or the root parts of the cotyledon, root parts of the seed. So this suggests that this this ATCLCs are present mainly in the chloroplasts that are forming or that are formed, and that's why you can see it in the green tissue. Now, what is the function of CLCE plants? In order to understand that, there was mutants that were developed from the ATCLCE, and the two mutants that were made were haplophytic mutants called CLCE2 and CLCE1, and the functionality of these mutants with respect to the wild type plants were assessed by using chlorophyll fluorescence. Also, the phenotypic traits of the CLCE mutants were checked in order to understand their localization, and overall, because these mutants are mutants of CLC, that's why they were the functionality of these chloroplasts were visualized with respect to that of the Y-type chloroplast. On highlight intensities that were induced on these samples, it was seen that there is a strongly polyphasic fluorescence time course, as you can see in the graph that is present on the right hand side. There was a reduction rate of the plastoquinone was calculated, and the reduction rate was actually faster than the quinol reoxidation rate, and that was the major reason why there is this polyphasic fluorescence time course that is observed in these samples. So there is an initial rising phase, and this initial rising phase that we see, the half time of this rising phase is similar in all these three different kinds of samples used, the wild type and the two CLC mutants. And the along with this result that is we see, also the quantum yield that was produced by these three types of samples were same. Now, why was the initial rate similar? And it could be because the light trapping efficiency of PS2 that was present was unaffected by the mutation of CLCE, and that's why we see similar initial rising phase. However, over time, the mutants displayed that the fluorescence increase component was slowed down around 10 millisecond time range, and around 100 millisecond time range, it's started becoming unaltered. So the two phases of disappearance that was seen was or the slowdown that was seen was due to the disappearance of the quencher concomitant with the reduction of plastoquinone pool, and with the reduction of soluble PS1 electron acceptors that are supposed to be there. This alternation of the kinetics of fluorescence that is observed in between the wild type and the mutant most possibly arises because of a variation in ionic strength. Although direct evidence is not present in case of Arabidopsis, but it could be assumed that due to ionic strength there could be any kind of impairment between the anionic permeability of the thylakoid membrane, and this could result in some kind of change in the thylakoid structure, which could result in a reduced plastoquinone pool that we see based on the graph present. and that could be the reason why we see such a polyphasic plot for looking into the chlorophyll fluorescence for the mutants versus the wild type. The next question was, where is ATCL-CF protein located? Now we know that ATCL-CE is present in the plastids. When the ATCL-CF proteins were tagged with GFP, the localization of these particles, when seen, these particles had a diameter of 1 micrometer, and uh, that suggested that these are not present in the plastids. And also, there was a co-expression of ATCL-CF GFP with SYTPDS red that was done similar to the experiment that was done for ATCL-CE, and the experimental results are present from panel A to C on this right hand figure. If you look at it, they are present distinct from each other, and that suggests that these particles are not present in the same location, which means that they are not present in mitochondria or they are not present in chloroplast. A similar kind of experiment was also done with another protein that is targeted to peroxisome called SKL22DS red, and that also had similar result, and that excluded from this protein being targeted to peroxisomes. There was a experiment that was done with a endocellular trafficking protein inhibitor called brephelidin A, and the results caused the aggregation of the particles and the redistribution of green fluorescence of the ATCL-CF GFP protein, which suggested there was an association with a BFA sensitive compartment for this experiment. And that means that since BFA could cause endocellular trafficking into Golgi, there could be a Golgi related targeting of this protein. Uh, in order to understand if this is present in Golgi compartments and if they are present in the cis Golgi compartment versus the trans Golgi compartments, experiments were done with a MAN99 fusion of GFP that could actually show the difference between the cis and the trans Golgi, as you can see in panel number D. Now, along with this MAN99 GFP, there was a co-localization experiment done with the red fluorescence particles that were ATCL-CF along with DS red 2, as you can see in panel E. And if they are co-localized, in that case, as you can see in F, you could see the yellow colored glowing particles of our co-localization, which suggests that yes, they are present in the Golgi network, and co-expression of ATCL-CF with protein targeted to trans-Golgi cisternae was also performed. And the results that were obtained by using the FUCT GFP, that actually would help it to be a marker only for trans-Golgi, actually suggested that generally this ATCL-CF would reside mainly to the early cis-Golgi subcompartment or to the Golgi compartment, but to the cis portion, only like the cis vesicles, and less to the extent to the trans Golgi cisternaes. So basically, these results and these experiments show that ATCL-CF protein is present in the Golgi vesicles. Now coming to the functionality of this ATCL-CF protein. We know that this is present in the Golgi vesicles, but we don't know what the role of it is. Now, in case of the CLC proteins that are present in yeast, also known as SCCLC, there is a mutant variety that is defective in growth, and it is generally defective in growth in presence of iron limited medium or in minimal medium, and also if there is any non-fermentable carbon sources. So in order to check the role of this ATCL-CF, ATCL-CF was inserted into the system of yeast that had the SCCLC in order to complement the functionality of the uh, yeast system, to see if the mutant yeast, that is GFP1, would grow or not grow. Now generally it has been seen that ATCL-CD and ATCL-C, which are also chloride ion channels, they would restore the effect that was caused due to the mutation, the GEF1 mutant variety in the yeast SCCLC, but not ATCL-CA. And that is why the authors decided to experiment on this system to see if ATCL-CF had any role in complementing the mutant, the growth mutant that we can see in case of the SCCLC, if it affects the growth. Uh, for this experiment, they chose two mutant strengths of the SCCLC. One was known as RGY86, which is a haploid, and another one called RGY192, which is a diploid. And in this experiment, the results that you can see on the right hand side, they are from the mutant strain RGY86, and similar results were obtained for actually both of the mutant strains. So basically, in the results that is shown over here through the panels, there were a bunch of experiments done with the wild type, with the mutant GF1, and then the GF1 complemented with various chloride ion channels such as CLC D, E, and F. Now, just to remind you again, CLCE is present in the chloroplast, whereas CLCF is present mainly in the Golgi. Now, the wild type can grow properly all across the different kind of systems that were provided to the organism. However, for GF1 mutant, it was unable to grow on non-fermentable carbon source that was not having the high iron concentration. And this growth defect was complemented in presence of CLCF, where you could see the growth, as you can see in between channels, in between the panels A and B. However, this effect was not present in case of CLCE, where you do not see the growth as visible from the panel B of the experiment. And the similar kind of effects you can also see in other cases where complementation of copper added to the media actually helped in the restoration of the growth defects that was present for this mutant. The reason why copper could allow the growth of the other genotypes was because most probably the intake of copper is helping in opening a complex called FET3-CC2 complex that helps in iron uptake from the background, and that could be regulating some of the growth, as you can see with the SD plus CU panel E and the panel G, SGE plus CU panel experiments for the mutants. However, that would be absent in the panels that didn't have the copper to that extent. Also, it was seen that the ATCLCD could restore the growth defect, and that could be because ATCLCD also co-localizes in Golgi and could have some effect in helping in restoring the growth defect. And one thing that from this experiment was clear, that CLCE is not competent enough in and actually not competent at all, to be honest, with in normal minimal media to restore the effect of growth that was seen in GEF1 mutants. So that is why ATCL-CF may be important in helping in the growth of organisms, and that seems to be the important functionality for ATCL-CF. So this brings me to the conclusion of this presentation, and from this paper, what I learned was there are 7 different types of CLC anion channels, and ATCL-CE and ATCL-CF were less studied, and that's why this was studied in this paper. And here they showed effectually where these 2 proteins are located. One is located in the chloroplast, and ATCL-CF is present in the Golgi vesicles, and that they have very different functionality, as shown by the experiments that were done using yeast complementation assay and the mutant assays. Overall, I think this paper was amazing, and the techniques that were discussed in this paper were overwhelming and really nice. I would suggest that uh, if you want, you should go through the techniques in order to learn how the techniques were done, because these are really good techniques, and it's pretty interesting to read this paper. I hope you enjoyed the presentation. Please feel free to leave your comments below and let me know about how I can improve things, how I can improve myself in presenting the articles, and how can I make this reading an article challenge much more better. So on the screen I have the chart for the days I have read the article. If you are reading an article and you are doing this 7-day challenge, you can definitely print screen of this uh, slide or this table, and or make a table of yourself to keep yourself motivated to read each day a paper, and in that way you will find your motivation to keep on reading papers. If you like this video, please put a thumbs up. If you don't like this video, also put a thumbs down. Every like or dislike is highly appreciated, and if you like this channel, subscribe to this channel to see more of such kind of contents. Thank you so much. Till tomorrow, have a good day.